Good evening all. We'll wait for 5 more minutes to let others join and then we'll start. Can you all see this in the slideshow mode?
Yes, ma'am.
Okay, yeah, thank you. So, welcome back to week 12 of PMRF TA sessions for the NPTEL online course Drug Delivery Principles and Engineering. So, we'll go over some of the key points discussed in this week lectures. So, in the last week, we have been talking a lot about these vaccines, in particular cancer vaccines, which can be of two types, prophylactic and therapeutics. So, in case of prophylactic cancer vaccines, we saw that there are some viruses which are capable of causing cancers in humans. So, to avoid getting those infections, this is what the prophylactic cancer vaccines are manufactured against. So, if you already deliver these cancer vaccines containing some part of antigen or some protein of this virus, then whenever there will be an actual infection from the virus, the body will be protected. So this is one example of prophylactic cancer vaccine. And on the other hand, then we have this therapeutic cancer vaccines, wherein you already know that a person has a cancer, a particular type of cancer, and you know that the patient's body is not able to overcome this cancer. So this particular therapeutic cancer vaccines are given in order to strengthen the immune system of that particular person against that cancer. So this is what we discussed in the last week. So this week we are going to we've seen some new concepts which are stimuli responsive drug delivery. So until now, so until now, whatever drug delivery systems we have seen, be it the reservoir systems or your hydrogel systems, these all were in a way where you deliver this particular drug delivery system and the drug keeps on being released from this particular system. So instead of that, we can have something known as stimuli responsive drug delivery. So the drug will not be released continuously from the systems but will be delivered in response to some stimuli, some signal coming from the system. So what will this give you? This will give you some control over how the drugs are delivered, how much amount of drugs are delivered, and at what time. So an example for this could be, so if you've seen diabetic patients, they usually take insulin after they have taken a meal. So the insulin doesn't have to be in their body for the entire time. It is required after taking a meal to lower down the blood sugar. So instead of just giving insulin as it is, what we can do, we can put insulin in a stimuli responsive drug delivery system, and the stimuli that the person has taken a meal could be a signal for the delivery system to then release the insulin. So it doesn't have to be in the body for the entire time. So this allows you easy switching on and off of that system. So to have the stimuli responsive drug delivery, it could use various different stimulus. It could be a magnetically controlled, it could be it could be ultrasound control, it could be pH responsive or temperature responsive, or you can make it a light sensitive or a ligand sensitive. So these are various kinds of stimuli which could be used to make the stimuli responsive drug delivery system. So we'll see some of these in detail. So first is pH responsive. So you must remember from our initial lectures, we have been talking about these enteric coatings. So we saw this in the module where we have discussed how drugs are administered orally. So in that case, what are these enteric coatings? So these enteric coatings are made of polymers. So you have your particular drug delivery system, you coat it with a polymer. That polymer now has a lot of carboxyl groups. So whenever you deliver these systems orally through the oral route, they are definitely going to land up in the stomach where the pH is very low. So at lower pH, what happens? These carboxylic groups, which are your COH groups, will be not will be protonated. They will be in COH form. So this means they are not soluble. So there is no charge on this. These are neutrally charged, the COH groups. So if there is no charge, they won't be much soluble. So your drug will not be released in the stomach. But when these particles pass from the stomach to the intestine has a very high pH compared to the stomach. At higher pH, what happens? All your COH groups will be deprotonated. It means there will be CO minus, H plus will be leaving. So now these polymers are charged. So charged polymers will definitely be more soluble in water. So what happens? These enteric coatings will then be soluble in water. In that process, they will then release the drug. So this is one example of having pH responsive polymers. Another example could be, let's say there are some polymers which, as I said, have a lot of high carboxylic groups. So if you make a polymer out of, if you make a drug delivery system out of such polymers which have many carboxylic groups, at lower pH, as I said, they'll be uncharged. So what happens at higher pH? They will become negatively charged. So let's say you make a hydrogel out of this polymer, and because now they are at lower pH, your COH groups are as COH, they are protonated and they are uncharged. So they will have a particular pore size, these hydrogels. So your drug is inside the hydrogel. Because the pore size is now small, your drug is not being released outside. But what happens when these hydrogens encounter a higher pH? Because of the higher pH, your COH groups will be protonated. They will be negatively charged. Now, if you have two CO minus groups nearby each other, they will definitely repel because they are both negatively charged. So what happens? They repel each other. In that process, they start swelling, and now the pore size of the hydrogel increases. So your drug can now be easily released from this system. So this is another example of pH responsive hydrogels. There are some other polymers which, so we have also talked about this proton sponge effect, right? Where once you deliver particles and they are taken inside the cell through endocytosis, they'll end up in through phagocytosis, they'll end up in the endosomal membrane, from where they'll then escape because of the presence of certain secondary and tertiary amines. So there are also other polymers which have this capacity of interacting with the endosomal membrane. So what happens? These polymers at lower pH, they will become more hydrophobic, and because they're hydrophobic, they will interact with the endosomal membrane and they will start disrupting this membrane. And in that process, they will then release all their cargo inside the cell. So this is another example of pH responsive polymers. The next topic to be discussed is temperature responsive hydrogel. So just like in pH responsive, the signal that is being given is coming from the change in pH. So for temperature responsive hydrogels, the signal is coming from the temperature difference. So in this particular drug delivery system, certain kind of polymers are used, which are known as LCST polymers. These are low critical solution temperature polymers. So what happens normally, most of the polymers, if you see, they have this property of at higher temperature they'll increase in its solubility. They will be more soluble in water at higher temperature. But these particular type of polymers, the LCST polymers, they have this property be less will be their solubility. So what happens is at higher temperature these hydrogens actually shrink, and at lower temperatures they swell. So this is another example of having stimuli responsive drug delivery. The next topic in this week's lecture was targeted drug delivery. So until now we have seen that whatever drug you're trying to give, you either give it orally or you inject it into the bloodstream, and then that drug will then go all over the body. So instead of that, if you want to say, let's say there's you're trying to treat a disease which mainly affects the lungs of a person. In that case, you want your drug to be mostly delivered into the lungs, and because you don't want it to go into the other parts, it has no function there. So this is known as targeted drug delivery, when you specifically target a given cell or a given organ. So this drug delivery targeting could be of two different types. It could be passive targeting or it could be active targeting. Now what happens in the passive targeting? You're not doing anything different to the drug delivery system. You're actually taking help of your body. So let's say we have already discussed this example, which is the EPR effect, which is enhanced permeation and retention effect. If you people remember, this happens mostly in case of tumors. So whenever there is a tumor in the body, the tumor cells are actually trying to feed a lot. They require a lot of nutrients. So they don't usually have a very organized structure. They're just grown haphazardly, and the blood vessels which are getting the nutrients there nearby the tumor area, they are actually very leaky in structure. So the endothelial cells which line the blood vessels are not very tightly packed, and they have a lot of gaps in between them. So whenever you deliver your micro or nanoparticles through the blood vessels, most of these particles will end up going to the tumor because of the leaky architecture of the blood vessels. So this is known as enhanced permeation effect. And also the tumor cells don't have a very good lymphatic drainage system. So whatever is being delivered to the tumor cells will actually be retained for a longer time in the tumor environment. So this is one example of passive targeting. So you have not done anything to the particles, but because of the architecture of how the tumor cells form, the whatever you have delivered, the cargo eventually lands up in the tumor cells. So this is one example of passive targeting. On the other hand, we have something known as active targeting. So here now we are going to modify your drug delivery system in such a way that it will go and target a particular organ or a particular tissue. Now what can you use for this targeting? You can use antigen-antibody interactions. For example, let's say there is this tumor in the body, and I know what kind of tumor it is, what kind of cancer cells are there in this tumor. And so what I'll do is, because I know what cancer cell these are, I also know that some proteins that are displayed on these cancer cells, I know what is the structure of these proteins. So what I can do, I can make my delivery system on the top of that, on the surface of these particles, my drug delivery particles. I'm going to attach certain antibodies which are complementary to the protein that is there on the cancer cell. Now, when you deliver these particles, since the antibodies that are present on your drug delivery system, they are going to identify these proteins, and they will bind only to these proteins. So it will not bind to any other protein that is present in the body. So they will not go anywhere else. They will be specifically targeting the cells, the cancer cells which have the protein which your antibody is complementary in structure. So this is one example of targeted drug delivery. Another is aptamers, making use of aptamers. Now, what are aptamers? You must have heard about this term before. So, aptamers are nothing but short oligonucleotide sequences. What is oligonucleotide? Oligonucleotide is nothing but DNA sequences, your ATGC sequences. Your DNA is actually a very big molecule. Instead of that, if you have some short oligonucleotide, which could be a 20 to 30 base pairs of ATGC sequences, these are known as your aptamers. You can make them, these are synthesized also. So what the one property these aptamers have is, because of the unique sequence that they have, they can actually fold themselves in a tertiary structure. They have a 3D structure. This is not just a single strand of DNA. These are actually 3D structures. Now these 3D structures are very, very specific to a particular kind of protein. Let's say, so you can actually synthesize aptamers the way you want them. Let's say you have a protein and you want an aptamer that specifically is has a shape that can be complementary to this protein. So you can then synthesize this. So what you can do, you can have your drug delivery system. You can attach aptamers on top of these drug delivery particles, just like you attach antibodies, and then you can deliver this. So now your aptamers are going to be very specific to certain proteins, so they will just go and bind to that. So this is another example of having targeted drug delivery. The next point that was discussed is nanotoxicity. So we have been talking a lot about these micro and nanoparticles, how they are very they could be very useful in treating a particular disease. They have a lot of advantages. But as all systems come with both advantages and disadvantages, one of the biggest disadvantage of having micro and nanoparticles is the toxicity associated with them. So if you have a macro system versus when you have something that is of a micro or nano range, because they are of a very small size, they get certain properties. Now, what property are micro and nanoparticles getting? Because these are very, very small in size, they actually have a larger surface area. So compared to a macro system, if you chop down this macro system in very small particles, you are actually exposing most of the area of these particles. So now, because the surface area of these net surface area of these micro and nanoparticles is very large, they interact with a lot of proteins that are present in our body. So this then leads to toxicity. So the toxicity that is associated with nanoparticles is actually known as nanotoxicity. It's a very famous field to work around, because as we have seen, we have a lot of advantages of this nano and micro particles, but because of this toxicity, they are not used properly in the we cannot use them until and unless we overcome this nanotoxicity. So to test whether your particles are going to create any toxicity, we have certain methods to do that. These could be a subcellular systems or your in vitro models, wherein let's say you have developed your formulation as a nanoparticle. To test whether it's going to be toxic or not, you can try incubating these particles with cell lines. Let's say I take some mammalian cell, for example, THP1 macrophages. I will then incubate my nanoparticles with the THP1 macrophages and monitor how whether it's changing the behavior of these cells or not, whether my cells are dying or not, whether they are metabolically active or not. So these are some of the experiments which could be done in the lab. done on isolated organs. For example, I take a mouse, I extract its liver, and then put my nanoparticles along with that liver and see what is happening to the liver cells. These are all your in vitro studies. Finally, when you see that no toxicity is occurring in the in vitro studies, I can take it up to the in vivo studies, where I take live mouse, I deliver my nanoparticles to the mouse and monitor the health of the mouse. I can take its weight, I can see for visual signs if it's having any problem, if there is inflammation in the body which is caused by the nanoparticles, and once I see that it's safe, then you can take the study further. So there is actually a proper protocol for taking for whatever research you do in your lab. From the lab to the market, there's actually a very big protocol. So this is where your clinical trials come into picture. So you must have heard a lot about these. Certain medicines are in phase 1 trials, certain drug delivery systems are in phase 2 trials. So what does this mean is, once you have done all your preliminary studies with mouse and you have established that my drug delivery system is very very efficacious, it has a nice efficacy against a particular disease, and it's also safe in vivo models, we can then go for clinical trials. So what happens in clinical trials? First you do your studies with mouse, then maybe you can take it to rabbits, then you can take it to bigger animals like sheep or horse, and then after that you can take it to non-human primates, which are your monkeys. You can test your particles in monkeys and see that if it has both efficacy as well as it's not toxic, and then after that you have to go for the human trials. So these clinical trials are all done with human patients. So they are actually divided in 3 phases. You first have phase 1, wherein you do it in a very small circle, let's say 10 to 15 patients, and you first assess whether your formulation is safe or not. If you see that it's safe, you then go to the phase 2. Phase 2 will also check whether a particular dosage of your particles is safe or not, and it also sees whether it is efficacious. Once your particles pass the phase 2 trials, then you can take it to phase 3, which then involves a lot of a bigger number of patients, and it is done all over different centers or different hospitals, and then you test your efficacy at the scale. Once you see that your drug delivery system has passed your phase 3 trials, then you get the FDA approval to go into the market. So this is how it stands. So these are some of the key points in this week lectures. So if there are no doubts here, we'll then move on towards some of the questions. Okay, let's start with the first question. So what are GMP and QS regulations? We have 4 options to choose from. So if anybody knows the answer, I'll pause for a minute and see if anybody has a response. If not, then we'll discuss the answer. Does anybody know the answer? Okay, no, nobody. Then I'll discuss the answer. The correct answer is GMP regulations apply to things which are intended to be used in humans is the correct answer. So what are GMP? GMP is nothing but good manufacturing practices. So these are protocols that are properly defined for, and these apply to anything that has to be used in humans, be it food or be it medicine. So you can read about more about this GMP and quality control regulations in this particular slide. Okay, then I'll move on towards the next question. Next is NIPAM is a temperature responsive hydrogel, and the following are its characteristics. So we have to choose one correct answer from this. Does anybody want to volunteer? I'm audible to all of you, right? Anyone wants to give it a try?
Their water solubility decreases with temperature.
Yes, that is one response. Thank you for answering. Anybody else? I guess not. Yeah, you're perfectly correct. We just discussed in the introductory slide where these temperature responsive hydrogels are made from particular polymers which are known as LCST, low critical solution temperature, and the one property they have is their water solubility actually decreases with increase in temperature. Clear? Okay, let's look at the next question. What happens during phase 3 clinical trials? And we have to select two options from this.
Ma'am, fourth one.
Okay, fourth, that is one response. Do you want to choose other option as well? Okay, somebody saying 2 and 3. Okay.
No, ma'am. Option 1 and 3.
Okay, option 1 and 3. Okay, I guess you're correct. So the correct answer for this is safe dosage is determined in phase 3 trials, we just discussed, right? So phase 3 trials are the last, are the most advanced of the clinical trials. So in this you first determine what is the dosage that is going to be safe for your human patients, and it is generally a multi-layer and multi-centered process. So it occurs all across the places, not just confined to one hospital center. You can coordinate with many patients that are present in various different places, and then that's how this process happens. Clear? Okay.
Ma'am, why not option 4?
Yeah, so as I said, these phase 3 clinical trials are usually done on human patients. So this last option, I think it is limited to monkeys. Studies are performed on large animals such as primates. So when you say primates here, these are non-human primates. These are basically your monkeys. So the monkey studies don't happen in phase clinical trials. These are usually done before you take your formulation to clinical trials. Is it okay?
Yes.
Yeah. Okay, let's look at the next question. So next is what is meant by sterilization.
Elimination of contamination.
Yeah, you are correct. Sterilization is elimination of contamination. I don't see anything much to discuss in this. So the concept of sterilization is basically removing all the living forms from your formulation. So yeah, elimination of contamination. Okay, the next question is which of the following are not examples of active targeting? And we have to choose two options.
Last two options.
Okay, EPR effect and direct local delivery to target organ. Okay.
Last two.
Okay, yeah, both of you are answering the same, and both of you are correct. The so both of these are passive targeting actually. So active targeting comes when you take help of some interactions like lectin carbohydrate or ligand receptor or antibody antigen. Your EPR effect and direct local delivery to target organ. So this could be your directly delivering particles to the lung or your intra-articular delivery wherein you deliver your drug delivery particles to directly into the joints. So both of these are passive targeting. You can read more about it in this slide. I'll move towards the next question. Next is what are the main disadvantages of aptamer? And again, two options.
They do not interact with positively charged targets.
Okay, that is one response. You want to choose another answer as well?
Can be detected by toll-like receptors.
Okay, you have chosen 1 and 1, 2, 3, 4. Anybody else wants to give it a try? Thank you for answering. No one? Okay.
And do not to interact with negatively charged.
Yeah, you're correct. So we are looking at disadvantages for aptamers. So aptamers, as I said, are DNA sequences. So they are negatively charged. So if you have a target which is already negatively charged, your aptamers are not going to be of much help there because they'll repel the negative charge. So aptamers are very good to target something that is positively charged. So one disadvantage is they do not interact with negatively charged targets, and also these can be detected by toll-like receptors. So your toll-like receptors are part of your immune system. They are trained to identify foreign DNA. So no matter, even if it's a DNA sequence, once you put it into a patient's body, the human body is going to consider this as a foreign particle and will try getting rid of your particles which display this aptamer. So these are two main disadvantages of aptamer. They do not interact with negatively charged targets and they can be detected by toll-like receptors. Okay. Let's look at the next question then. Next is which of the following statements is false for enteric coatings? One correct answer.
allows delivery of drug in the stomach.
Okay, 1, 2, 3, 4. You have chosen option 4. Thank you for answering. Anybody else wants to give it a try? No? Okay. Yeah, you're perfectly correct. It allows delivery of drugs in the stomach. Actually falls for enteric coating. So we just discussed this, right? So enteric coatings are made of polymers which have a lot of carboxylic acid. So once you deliver it orally, it will go to stomach, which has a very low pH of somewhere around 2. So at this pH, all your carboxylic acids will be protonated. They will be COH form, and hence they'll have low solubility. Once these particles go to small intestine, which will have a pH of around 8, all your carboxylic acids will be deprotonated, and this will allow it to be soluble in the water, and that's how the drug gets released in the intestine. So if you look at the options, pH responsive, we have to select for the false statement. So pH responsive, definitely enteric coatings are pH responsive. They are insoluble at low pH and soluble at physiological pH. They protect acid-sensitive drugs from gastric juices, deliver drugs to small intestine, and can be made of polycarboxylic acids are all true. They allow delivery of drugs in the stomach is the only false sentence here. Okay. Let's look at the next question. Next is PTEAAM is widely used, it's a polymer, is widely used as a temperature responsive hydrogel because of its LCST in the range of
25 degrees to 32 degrees.
55.
I can hear two responses. Somebody answered option 1. The second was anybody else trying to answer? Okay, yeah.
Now,
Yeah, I think both of you are answering the same option. So yeah, you are correct. This particular polymer has LCST in the range of 25 to 32 degrees Celsius, which it's a pretty straightforward question actually, nothing to discuss. You can get this information in the slides as well. Okay. Next is which of the following functional groups would make coating non-liable to acid damage in stomach? Seems a little repetitive. Sorry? Can you please?
Ester group.
Okay. Yeah, option 1. Okay. Anybody else? Thank you for answering. Anybody trying to answer before I discuss it? Okay. The correct answer is actually carboxylic acid groups. This is what we have been discussing, right? Even in the enteric coating. So something we need functional groups which would make the coating non-liable to acid damage in stomach. So that is done by your carboxylic acid groups, because at the pH in the stomach, all your carboxylic acid groups will be insoluble. So they will protect your drug which is encapsulated into these polymers. Is it clear? Okay. The next question, this is the last question for the assignment. What assays can be conducted to test for nanoparticle toxicity in vitro? And we have to select two options.
Transcription
Okay. Do you want to choose?
and cell metabolism.
Okay, you've chosen the last two options. Thank you for answering. 2 and 3. Okay. Anybody else has any response? Thanks for answering both of you. Okay, let's look at the answers. So the correct answer is 2 and 3. I guess one of you answered correctly. So hemolysis assays are actually done to see whether so sometimes what happens is these nanoparticles being of a very small size and they are also very interactive with the proteins present in the blood, they actually go and attach to they can cause lysis of your RBCs. So what you can do is to check whether your nanoparticle is going to lyse your RBCs. You can incubate these. You can collect blood from an animal, from let's say a mouse, and you can put these nanoparticles in the blood and see what happens to the blood over time and see whether your RBCs are getting ruptured or not. So that's what is done in hemolysis assays. And then cell metabolism assays are, as I told you, you can take THP1 macrophages or any other cell line and see what is the effect of your nanoparticles on these cells, whether they are metabolically active, whether they are dying fast or their morphology is changing, and so on. So these two assays are done to test nanoparticle toxicity in vitro. Is this clear? Any doubts in this? I think that's all I have for you. Yeah.
I have a question.
Yes.
Like, can we do transcription to check the toxicity of the nanoparticles?
Okay. Can you tell me what is transfection?
Transcription is introduction of any plasmid or any DNA to mammalian cell line.
Yeah, so that's what transfection is, right? So let's say you're doing gene therapy, you put a DNA in the mammalian cells. So that is not going to tell you whether so this is a completely different concept than your nanoparticle. There's no delivery of nanoparticle happening here, right? So whether your DNA is getting into the mammalian cells or not is not going to tell you whether whatever you have delivered is toxic to the cells or not. Makes sense?
Yes.
Yeah.
Thank you.
Yeah. Thank you. So that's all I have for you for this assignment, the 10 questions. If you have any other topic that you think we should be discussing, we can do that. I'll have one more lecture next Monday. This is like an extra lecture apart from these 12 assignments. So in that we can discuss some of the numericals because I did not discuss any numericals in all the 12 lectures, not much. So in that we can discuss the numericals which could be asked from the drug delivery aspect in your final exam, or any other concepts which you want me to repeat in that will be the last lecture, the next Monday lecture. So if you guys have any doubts, you can ask me now, and we could try clearing it up in the next lecture as well. Or if you can just go through most of the material, and if you come up with some questions, we could discuss that as well. Thank you, Soumya. So is there any other topic you would like me to go through? If not, then we can stop this meeting and I can meet you people next Monday. Is that all? Shall I stop the meeting, or the rest who are there in the meeting, do you have any questions, any doubts? Okay, thank you. I hope I'm saying your name correctly. I don't know how to pronounce that. Okay, thank you. See you people next Monday then. Bye.